Boarfish, Capros aper, are a small, laterally compressed, well-armored species. They have between 9 and 10 dorsal spines, 23 to 25 dorsal soft rays, 3 anal spines, 22 to 24 anal soft rays, and 21 to 23 vertebrae. They have large bulging eyes and a snout that can grow as long as their eyes or even longer. Their mouth is very protractile and forms a short tube when protruded. Dorsal and anal spines are only a little longer and stronger than the others, and their body depth is more than 1.5 times its standard length. Boarfish are gregarious. They form schools and inhabit a depth range of 40 to 700 meters in pelagic and demersal waters. They aggregate around the shelf edges and in areas of high productivity. Their distribution ranges along the eastern Atlantic, western Norway near Skagerrak, in the Shetlands, along western Scotland to Senegal, and in the western Mediterranean. In the northeast Atlantic, spawning aggregations form off the south coast of Ireland from June to August. Their overall abundance appears to be increasing in most regions like the Celtic Sea and the Bay of Biscay. Hypotheses for the increase include climate change or variability in their distribution. Recently, it has been discovered off the Portuguese coast that the species can be classified into two distinct morphological types, with some individuals having intermediate characteristics. One of these types has smaller eyes, a deeper body, and a more concave supraorbital margin than the other. This observation has led to the hypothesis that the current systematic classification of boarfish may be mixing different species. Results from multidimensional scaling analysis is showing that there should be a differentiation between the two morphotypes into species of Capros. The existence of distinct forms of boarfish has not previously been reported, which is odd given that this widely distributed species has been sampled thousands of times during bottom trawl surveys along the Portuguese coast. This is probably due to the fact that boarfish, in one haul, belong to the same morphotype, as each morphotype tend to aggregate separately and as a result are landed apart. Apparent differences in the reproductive characteristics in the Northeast Atlantic region and within Portuguese waters have been reported. However, reproductive isolation between morphotypes along the Portuguese coast is unconfirmed. Body shape is determined by several factors, including genetic pool, developmental constraints, responses to environmental influences on embryonic and larval development, and natural selection on adults. Historically, boarfish concentrations in the Northeast Atlantic were considered rare. However, an increase has been observed, and in more recent years they have been described as nuisance bycatch in mackerel fisheries. Nowadays, spawning aggregations have become the target of commercial fishmeal fisheries off the west coast of Ireland by Irish and Danish vessels. Fishmeal plants in Denmark have recently been replacing sand eel with boarfish, and this is a concern as little information exists on boarfish life history, which is a necessity to manage and assess the potential damage this fishery could have on the stocks. Increases in abundance of boarfish in this area have been attributed to enhanced adult growth and recruitment as a result of climate-related changes in environmental conditions. The relatively new development of the trawling fishery for boarfish came as a result of this increase. Boarfish in the Northeast Atlantic area are long-lived species that reach an estimated age of 26 to 30 years old, with a late age at maturity of roughly 5 years. They have relatively fast growth rates of 0.186 per year and a total length at maturity of 9.7 centimeters. Natural mortality estimations of boarfish is at a level of 0.25, which is relatively low, presumably due to the spiny nature of boarfish. No seasonal effects occur in size distribution and sex ratio, indicating that females and males stay longer in shoals throughout the year. They are sexually dimorphic, where females are larger than the males, which is potentially a result of differences in maturation pattern, growth pattern, asymptotic size, or longevity between sexes. Both sexes have been shown to differ in terms of asymptotic length, with females averaging 130 millimeters and males 110. This asymptotic length is a parameter of the von Bertalanffy growth function, which expresses the mean length that a fish of a given stock would reach if they were to grow for an infinitely long period. This indicates that for boarfish, reaching asymptotic size relatively quickly is optimal, and surplus energy is then allocated to other life history traits, for example reproduction. Once the males have attained a size suitable for successful reproduction, they travel to the spawning grounds earlier and remain there for longer than the females. Therefore, males invest more energy into the reproduction. However, this act exposes males to prolonged fishing pressure and thus increased mortality, leaving fewer males in the population over 15 centimeters. This lower average age than females could also be explained by a hormone that increases swimming speed in the males and could lead to overexhaustion, not just a result of increased exposure to fishing. Boarfish invest a lot in its heavy armor, with a large proportion of energy spent on their dorsal and pelvic fin spines, spinules, barba, and barblets. It is evident that a suite of strong protective armor is a crucial life history trait for this small species. Heavy armor makes boarfish a less attractive food source for many species, however they do contribute to the diet of rays, sharks, and seabirds. This trade-off could potentially represent a key factor in allowing for redirection of most energy away from the soma to the gonads, following the onset of maturity and into considerable longevity. The quantitative genetic character longevity of a boarfish represents a more adaptive and optional life history strategy, where a high variability in pre-recruit survival is safeguarded by a supply of fish distributed across a wide range of ages. Infrequent directive fishing on this species has not yet caused the age truncation typical of long fish species, where fish can live long but are rarely found at such an age. However, it is a possibility. Due to its small size and a late age at maturity, boarfish is sensitive to high fishing pressures, even though their growth rate is similar to other pelagic species. This may be a crucial life history strategy of boarfish that could play a key role in our understanding of the potential impacts the fish meal fisheries could cause on boarfish stocks. Life history strategies, therefore, are a vital element in successful fisheries management, and with little work being done on this species, future research into their life histories will be necessary to ensure sustainability within the stock. The COI or CO1 barcoding gene has been used for various studies as species identification tools. Along with the COI, microsatellite genotyping by sequencing using individual combinatorial barcoding has been used to assess the genetic population structure of boarfish across the species range. Microsatellite loci were developed de novo and genotyped by next-generation sequencing. Genetic analysis of the samples indicated that boarfish can be subdivided into at least 7 biological units or populations across the species range. Understanding population genetics is tied closely to evolution, and understanding genotypic and phenotypic variation in relation to natural selection, as this is a driving force in population genetic variation. By developing a de novo suite of informative microsatellite markers for boarfish, biological populations were determined throughout the boarfish range to delineate the stock boundaries for a more specific management approach. Genetic markers were also used in this way to attempt to decipher if the recent increases in abundance of boarfish in the Northeast Atlantic area are a result of individuals relocating from other regions. By developing biological populations through genetics and mapping them, it is clear which areas are reproductively connected. Such a discovery is useful for stomach content analysis. It is not possible to determine from such a study that boarfish feed on the same prey. However, it is reasonable to infer individuals with the same genetics and spawning areas could also be feeding on similar prey. Genetics, therefore, can be extremely useful in the management of stocks, as it provides insight into life history strategies and biological characteristics, such as stock delineation, movement of the stocks, along with longevity, age of first reproduction, and even the lifetime reproductive output of a species. In conclusion, the study of boarfish life history traits is a relatively recent venture, which is indicative of the limited amount of information available. However, estimations have been made relating to the maximum age, their growth rates, age at maturity, and length at maturity. It has been determined that they are sexually dimorphic species, with females growing larger than males. Trade-offs have been noted between boarfish growth, reproduction, and protection via their strong armor and therefore longevity. Genetics have been used to delineate boarfish stocks into biological populations. However, more research into life history traits using genetics should be completed.